Hello, my name is Joshua Mugabe. I welcome you to protein synthesis. What is the genetic code? Genetic code is the key for the conversion of DNA nucleotide sequence into amino acid sequence that will compose proteins. Which is the biological knowledge that contains genetic information that is transmitted hereditarily and controls the cellular functioning. The hereditary knowledge that controls the unicellular functioning is the DNA, deoxyribonucleic acid. The DNA contains information for protein synthesis in cells. How are the concepts of DNA, gene, proteins, and characteristics of living being related? Characteristics of an organism depends on the chemical reactions that occur in them. These reactions are catalyzed by enzymes, highly specialized proteins. Every protein of an organism is made from information contained in RNA molecules that are made according to a template sequence of nucleotides of DNA chain. A gene is a DNA polynucleotide sequence that contains information for the production of a protein. What is the role of messenger RNA and ribosomes for the protein synthesis? The messenger RNA is produced within the cellular nucleus and migrates to the cytoplasm, where associated with the ribosome, it guides the building of amino acid sequence that will compose proteins. Ribosomes are sites for meeting and binding of messenger RNA and transfer RNA. They are structures where amino acid transported by transfer RNA are united by peptide bonds, forming polypeptide chains or proteins. Of what subunit are ribosomes made? Ribosomes are made of two subunits, smaller subunit and large subunits. These subunits are made of ribosomic RNA, rRNA, and proteins. Ribosomes have 3 binding sites, one for messenger RNA and 2 for transfer RNA. How different are the location of ribosomes in eukaryotic and in prokaryotic cells? In prokaryotic, ribosomes are found free in the cytoplasm. In eukaryotic cells, they can be found free in the cytoplasm and mainly adhered to the external membrane of the carytheca and of the endoplasmic reticulum, the rough endoplasmic reticulum. How is the finding of ribosome inside the mitochondria and chloroplast explained? It is a strong hypothesis that mitochondria and chloroplasts were prokaryotes that associated to primitive eukaryotic cells under mutualism, gaining protection and offering energy. This explains why within those organelles there are DNA and protein synthesis machinery, including ribosomes. This hypothesis is known as the endosymbiotic hypothesis on the origin of the mitochondria and the chloroplasts. What are some examples of human cells that produce proteins for exportation? Which cytoplasmic organelle is expected to be well developed and abundant in these cells? Specialized cells of the glands, like Langerhans cells of the pancreas that produces insulin, or the saliva-producing cells, they are example of secretory cells. In specialized cells in secretion, the endoplasmic reticulum and the Golgi apparatus are well developed, since they participate in the storage and processing of proteins for exportation. Which are the more abundant ribosomes in secretory cells? Free cytoplasmic ribosomes or those associated with the rough endoplasmic reticulum? Free cytoplasmic ribosomes are more related to protein production for internal cellular consumption, while those adhered to the rough endoplasmic reticulum are more important in protein synthesis for exportation. Proteins made by adhered ribosomes enter the rough endoplasmic reticulum and later transferred to the Golgi apparatus. So in secretory cell ribosomes adhered to the endoplasmic reticulum are more notable. Where in eukaryotic cell does messenger RNA synthesis occur? Where do these molecules migrate? Messenger RNA molecules are synthesized in the nucleus. They pass through the pores of the nuclear membrane and gain cytoplasm to reach the ribosomes where protein synthesis occurs. After the fact that it is based on information from messenger RNA, what is the process of protein synthesis called? Protein synthesis is called translation of genetic information into proteins. What is the difference between transcription and translation? Transcription is the name given to the information of DNA molecules from an open DNA chain used as a template. Translation is the making of polypeptides. Those are amino acid bound in sequence and thus protein based on information encoded in the messenger RNA molecule. In eukaryotic cell, transcription occurs in the nucleus and translation occurs in the ribosomes. Transcription precedes translation. How do nucleotides of messenger RNA chains encode information for the formation of the amino acid sequence of protein? There are only 4 types of nucleotide containing bases that compose RNA nucleotides. We have adenine, uracil, guanine, and cytosine. Amino acids, however, are 20 different ones. Considering only one nucleotide at 1:1 coding, it would be impossible to codify all the amino acids. Considering 2 nucleotides, there would be an arrangement of 4 elements, 2 times 2, resulting in a total of only 16 possible codifier units, 4 by 4. Nature may know combinatory analysis since it makes the genetic code by arrangement of the 4 RNA bases, 3 by 3, providing 64 different triplets, 4 by 4 by 4. 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 4 by 4 by So each triplet of the nitrogen-containing base of RNA codify one amino acid of a protein. As this triplet appear in sequence in the messenger RNA molecule, sequential amino acids codified by them are bound together to make polypeptide chains. For example, uracil sequence codifies the amino acid phenylalanine, as well as uracil cytosine codifies the adenine cytosine uracil, adenine cytosine cytosine, adenine cytosine adenine, and adenine cytosine guanine sequences. They codify the amino acid threonine, and so on for all possible triplet sequences and all other amino acids. What is the name of an RNA sequence that codifies one amino acid? Each sequence of 3 nitrogen-containing bases of RNA that codifies one amino acid is called a codon. The codon is the codifier unit of the genetic code. Since among the 64 codons of the messenger RNA, 61 codifies amino acids that form polypeptide chains, what are the functions of the 3 remaining codons? Since there are 20 amino acids and 46 possible of messenger RNA codons, it is expected that some amino acids to be codified by more than one codon. And that really happens. Not all the 64 codons, however, codify amino acids. 3 of them, uracil adenine adenine, uracil guanine adenine, and uracil guanine adenine guanine, work on information that the last amino acid of the polypeptide chain under the production was already bound. That is, they signal the end of the polypeptide synthesis. These codons are called terminocodons. The codon adenine uracil guanine codifies the amino acid methionine, and at the same time it signals the beginning of the synthesis of the polypeptide chain. It is an initialization codon. In prokaryotic cells, there is a sequence called Shine-Dalgono sequence. In general, adenine, guanine, guanine, adenine, guanine, guanine, in the position that antecedes the initialization codon adenine uracil guanine. The function of this sequence is distinctness between initialization adenine uracil guanine and the other adenine uracil guanine codons of the RNA. What is the cellular structure to which messenger RNA molecule binds to start the protein synthesis? To make proteins, messenger RNA molecule necessarily associates to ribosomes. Ribosomes have two sites for the binding of two neighboring messenger RNA codons and where anticodons of transfer RNA bind by hydrogen bonds. Thus, ribosomes are the structure responsible for positioning and exposure of messenger RNA codons to be translated. In ribosomes, the peptide bond between the two amino acids brought by transfer RNA molecules also occur. The peptide bond happens when transfer RNA carrying amino acids are bound to exposed messenger RNA codons. How are amino acids brought to the cellular site where translation takes place? What is an anticodon? Amino acids are brought to the ribosome by RNA molecule known as transfer RNA or tRNA. One tRNA associated to its specific amino acid binds by a special sequence of 3 nucleotides to the messenger RNA codon exposed in the ribosomes. This sequence in the transfer RNA is known as anticodon. The transfer RNA anticodon must be complementary to the messenger RNA codon, to which it binds according to the rule of adenine with uracil, cytosine with guanine. Ribosomes then slide along the messenger RNA molecule, a process called translocation, to expose the following codon to the binding of other transfer RNA. When amino acids corresponding to the neighboring codons bind by peptide bonds, the first transfer RNA is liberated. Why is the proximity between ribosomes and amino acids important for the protein formation? What is the enzyme that catalyzes that reaction? The proximity between ribosomes and amino acid is important because the enzyme that catalyzes the peptide bond resides in ribosomes. As substrates of these enzymes, amino acids need to bind to the enzyme activation centers. The enzyme that catalyzes the peptide bond is the peptidyl transferase. Why do ribosomes move along messenger RNA during translation? During translation, ribosomes always expose two messenger RNA codons to be translated by moving along the messenger RNA. When peptide bond is made, the ribosome moves to expose the next codon. This moving is called ribosomal translocation. In the rough endoplasmic reticulum, ribosomes are attached outside the membrane and messenger RNA molecule, rather moving through them. How many of the same proteins are made at the same time by each ribosome in the translation of one messenger RNA molecule? How does consecutive protein production occur in translation? Ribosomes do not make several different proteins simultaneously. They make them one after another. Along one single messenger RNA molecule, however, many ribosomes may move in a real mass, manufacturing the same protein. The unit made of many ribosomes working upon the same messenger RNA molecule is called polysome. And messenger RNA molecule codifies only one type of protein. Eukaryotic cells have monocistronic messenger RNA. That is, each messenger RNA codifies only one polypeptide chain. Prokaryotes can present polycistronic messenger RNA. At the end of the assembly of amino acids into polypeptide chain, the messenger RNA, by one of its terminal codons, signals to the ribosome that the polypeptide chain is complete. The ribosome then liberates the produced protein. In prokaryote, after this conclusion, the information for the beginning of the synthesis of another different protein may follow in the same messenger RNA. If a transfer RNA anticodon is cyanine, adenine, adenine, what is it corresponding messenger RNA codon? For the genetic code, which amino acid does this codon codifies? According to the adenine, uracil, and cytosine guanine rule, the corresponding codon for cytosine adenine adenine anticodon is guanine uracil uracil. The genetic code table for translation is related to codons and not anticodons. The amino acid codified by guanine uracil uracil according to the genetic code is valine. If a fragment of nucleic acid has a nucleotide sequence, thymine, adenine, cytosine, can one assert that it is a codon or an anticodon? A nucleic acid having thymine, adenine, cytosine sequence surely is not transfer RNA. It is DNA. Since RNA does not present a nitrogen-containing base, thymine, since it is not RNA, it cannot be a codon or an anticodon. Why can the genetic code be qualified as a degenerate code? The genetic code is a degenerate code because they are amino acids codified by more than one type of codon. It's not a system in which each element is codified by only one codifying unit. For example, amino acid arginine is codified by 6 codons: cytosine, guanine, uracil, cytosine, guanine, cytosine, cytosine, guanine, adenine, cytosine, guanine, guanine, cytosine, guanine, adenine, guanine, adenine, adenine, guanine, guanine. What is the concept of universality of the genetic code? What are the exceptions to this universality? The genetic code is universal because the rules of protein codification based on messenger RNA codons are practically the same for all known living beings. For example, the genetic code is the same for humans, for bacteria, and for invertebrates. protein synthesis in the mitochondria and chloroplast, and some protozoans, however, are accomplished by different genetic codification. How does the universality of the genetic code make recombinant DNA technology possible? The universality of the genetic code refers to the fact that all living beings have their protein synthesis machinery functioning according to the same principle of storage, transmission, and recognition of information, including translation of messenger RNA codons. In fact, makes possible the exchanging of genes and gene fragments between different organisms and secure that these genes they continue to command protein synthesis. This universality, for example, makes feasible the insertion of a fragment of human DNA containing a gene for the production of a given protein into the genetic material of bacteria. Since the bacteria transcription and translation system works in the same manner as the correspondent human system do, the bacteria will begin to synthesize the human protein related to the inserted DNA fragment. There are industries that produce human insulin for use by diabetic patients. In this way, they synthesized by bacteria with modified DNA. If the genetic code was not universal, this kind of genetic manipulation would be impossible and very difficult to accomplish without the new technology progresses. Thank you very much for participating in protein synthesis. Please do like, share, and subscribe.